Welcome dear student in the first session of the Biology Practical. So in this Corona period, whatever the available is there in my hand, I am trying to show you the practical. Based on that, we'll start with the first practical, that is about the potato osmoscope. So the osmosis which is taking place in this. So the first experiment, that is study of the osmosis by a potato osmoscope. Okay, so before starting this, we'll just see the what is osmosis. So in the theory you learn in detail, that time you'll come to know. So what is osmosis? Osmosis is a type of the diffusion. So diffusion means what? That is a molecule or the atoms which move from the area of higher concentration to the area of the lower concentration. When the substance crosses the semi-permeable membrane in order to balance the concentration of another substance. So in this experiment, the potato acts as a semi-permeable membrane. So in this experiment, what are the requirements are there, that we'll see. So whatever the material is available in my hand, accordingly we'll do the experiment. So for this, first, that is about the potato, one big size potato you have to take. Second to this, you have to take the either the peeler or the scalpel. So I have the peeler at my home, so which I will use. Next to that, you have to get the knife. So here I have taken the knife, as well as I have the cutter also. So with the help of the cutter also, we can use this. Next to that, we require the pins. So this pins or the safety pin which your mother or your sister use, that you can use it. So whatever available according to that. Next to that, we require the petri dish. So it is there in the lab, but now what the plate I am having. So with the help of the plate. Another one, we require the water. So here plain tap water will take. Okay, then next to that, we require the color solution. So here, instead of getting the color, I have used the beet. So with the help of the beet, I have made the color, so that after the experiment, this potato I can use it for the making vegetables. So I don't like to waste the material. So okay, so instead of using the artificial color, you can use the natural color which is of the beet, or you can use the haldi also, or any other natural color. One more thing is required, that is about the sugar solution. So this sugar solution is already made here with the concentration 20% concentration. So half teaspoon of the sugar is mixed in this half bowl of the water, so which is required. We require the spoon also, just pouring this sugar solution in the osmoscope. So what are the requirements? Once again revise this. First one, that is a one big size potato. To peel off this, the peeler is required. To make the hollow cavity inside, either the knife or the cutter is used. So mark the level of this sugar solution inside. 2 to 3 pins you have to take. Then sugar solution which is of the 20%. Half teaspoon of the sugar is added in the half bowl of the water. Then the plain water which is from the tap we can take. Then next, the color, we want color solution. So here with the natural color which is made by the peat. Now we'll start the experiment. First you have to cut the potato in the sense, first you have to remove its skin with the help of the peeler. So here I am removing its skin. Why to remove the skin? Because if you keep the skin, it will not be permeable to water molecules to travel. This osmoscope, potato osmoscope, we are keeping in the water, so water molecules should get travel inside the potato osmoscope. So here, like that, you have to remove its skin with the help of the peeler. So in the regular practical also I inform all the students, especially to the boys, you have to keep the practice of using this devices, knife as well as a peeler, etc. Because after the marriage you have to help your wife. Now also you can help your sister and your mother, at least in this period. Okay, now we'll come to the point. This potato is peeled off, so you have to make its base flat so that it will stand properly on the plate. So here its base is made flat, as well as you cut from the other side. Now this way it should get stand. Okay, now I will make its potato, will make the inner portion hollow so that we can pour the sugar solution in that. Now with the help of the knife you have to cut carefully so that you can make the proper cavity. Care you have to take, it should not have any cut at the base or from the side, otherwise your whole process will get spoiled. So like that, here slowly you have to remove its inner portion and make it hollow. So go up to the nearest to the base. Base is already flat. You have to make the base thin so that water can easily travel through this. Don't be hurry while doing this, because in exam time you'll get only one potato. This practical is there, that is of the 3 marks. It will be asked in the question number 2. There is no option for this. Compulsory, whatever the experiment you are getting in the exam time, you have to perform that. Be careful while removing this inner portion. So avoid losing the marks. Another question also will be there, but X student will get one experiment, Y student will get the another experiment. So here carefully we'll remove so like that. Make it so hollow cavity. this wall of the potato, which will act as a semi-permeable membrane through which water molecules will travel inside that osmoscope. We will pour the sugar solution. This sugar solution, which is of the higher concentration, and this potato osmoscope we are going to keep inside the colored water. So of course water, this will having the lower concentration, so here now my potato osmoscope is ready. Inside this you can see clearly the hollow cavity. This way the inside hollow cavity you have to make it. It should not have any cut from anywhere. Okay, base should be intact. Now this is the osmoscope is ready. Okay, now second to this, what we have to do? Second to this, we have to take the water. We'll take the water in the petri dish. So here I have the plate. So in the plate I will pour the water. So half fill of water. In this I will pour the color of the beet. So now this is my colored water. Okay, now what I have to do, I have to pour the sugar solution inside this osmoscope. So with the help of the spoon I will pour this sugar solution. This sugar solution is there, which is of 20%. So concentrated it is. So this I will pour inside slowly. Now this I have to fill half. So it is half filled. You can see, and I will mark the level where this sugar level present, sugar solution is present. So here it is marked, you can see. Okay, now this I have to keep it in this plate where the color solution is present. Okay, now this is a experiment is ready. You have to keep it for the 2 hours, and after 2 hours we'll observe what is the effect, and then you can find out the conclusion regarding the same. So what actually is happening, just now I said here, this potato osmoscope, it's a whatever the wall is there, which act as a semi-permeable membrane. Inside this the sugar solution is there, which is a concentrated sugar solution. Outside, which is a water, and it is a color solution. So easily we can recognize when the color will travel inside. So water molecule goes from the region of lower concentration to the higher concentration. So inside it is higher concentration. So slowly water will travel inside, water molecules will travel inside, and the level of this sugar solution inside the osmoscope will rise. So this will observe after half hour. Okay, so till that time we'll just discuss regarding the question answers. Okay, Now we'll see about what you have to write in your practical book. Mention the date, so on which day I will upload the video. On the same day you have to make this experiment at home. I will revise once again what is required here. Main, that is about the potato, fresh potato you have to take of big size. Then use peeler, so peeler is there at your home. Then take the knife also. Instead of petri dish, you take any plate or bowl. Then you have to take the pin, any pin you take it. Okay. Concentrated sugar solution, so which is of 20%. So half teaspoon of the sugar, add in the half bowl of the water. And color water you can make it with the help of the beet, or we can help with the help of the haldi, or you can use your color from the color box anyway. So experiment, just now I showed how to perform. This is how we perform the experiment. This is now initial level. This pin is put it here inside where the level is present. About this color solution is there. Then inside concentrated solution of sugar. Now this plate is there where the color solution is present. After 2 hours, this level of the water, sugar solution which is present inside will rise up till the concentration will be balanced on the both the place, outside the osmoscope as well as the inside the osmoscope. Sometimes it get overflow also. So after 2 hours you have to check where the level is there, then insert the another pin here, then you'll come to know how much water the water is travel inside. Okay, so this is about what? About the experiment on about the osmosis using the potato osmoscope. Here what inference you'll get through this? Rise in the level of sugar solution and the presence of color solution in the potato osmoscope indicates. So after 2 hours you'll come to know. What it indicates that the potato osmoscope wall is there, which act as a semi-permeable membrane, and due to which the water travel from the petri dish or from the plate to the inside the cavity of the potato osmoscope. Okay. Next question, potato is used to prepare an osmoscope. Why potato is used as a osmoscope? Potato osmoscope, also called as the osmometer. In this, potato tuber is used. Water molecule move from the lower concentration to the higher concentration solution through the semi-permeable membrane of the potato cell. So that's why potato is used. So potato wall of this osmoscope which act as a semi-permeable membrane. What is the permeability? Ability of the membrane to permit the passage of substance is called as the permeability. It means this wall of the potato, what is the osmoscope is there, which permit that water molecules to travel. That is called as the permeability. Osmotic pressure. So osmotic pressure of the solution is the pressure which must be applied to the solution in order to prevent the entry of the solvent due to the osmosis. So this wall prevent the entry of the solvent here when it get balanced. So that is a osmotic pressure. Once it will get balanced, no water molecule will travel inside. It will remain there only. Do you find any role of the osmosis in water purification? So here we know that RO water, that is reverse osmosis, is a water purification process that use a partially permeable membrane. The partial permeable membrane is used to remove the ions, unwanted molecules, large particles from the drinking water. So here RO water we get, that is what? Fully purified. And that is by the process of what? Same. That is by the process of the osmosis. Okay. Next, osmosis play an important role in the stomatal movement. Explain. So opening and the closing of the stomata, which based on the turgidity of that guard cells. So the stomata is there. This stomata when open and when it get closed, it is related to that. So if you'll see in the diagram, the stomata, this is about the guard cells. Water get travel inside. As a result, this guard cell become a turgid. And when water travel from inside to outside, that time the guard cell become a flaccid. So this turgidity and the flaccidity is there. When it is a turgid, that time the endosmosis takes place. Endosmosis means going inside and exosmosis means coming outside. So here, during the daytime, guard cells become a turgid due to endosmosis. Water comes inside the guard cells. And during night time, guard cells become a flaccid. Water goes in the outside. That makes the exosmosis. And as a result, the stomata get closed and open. Yes, here you can see the rise in level of the water in the osmoscope. If we'll check inside this, you got the little bit color also, and the level of the sugar solution is rise up. So this we can put with the another pin at this place where it is rise. So this is checked after 2 hours. Okay, you can see the level is raised. So this is about the potato osmoscope, where the wall of the osmoscope, which act as a semi-permeable membrane, where the osmosis has taken place.